cycle. The TCA cycle generates NADH for oxidative phosphorylation and also important metabolites, amino acids, and other metabolites. And then we have oxidative phosphorylation, which generates ATP. Now, a lot of work has been done in my lab and in other labs showing that oxidative phosphorylation is perturbed in cardiolipin-deficient cells. Just to summarize the highlights, we know that there is a decreased stability of supercomplexes, perturbation of respiratory control, and then the sequelae from that, a decreased membrane potential, and decreased respiratory enzyme activity. But I'm not going to talk about OXPHOS today. What I'd like to talk about, though, is the TCA cycle and acetyl-CoA in our studies that show that both pathways are perturbed in cardiolipin-deficient cells. So I have three sort of sub-themes. They're not really independent. Everything is sort of interdependent, but for the purposes of simplicity, I'd like to talk about iron-sulfur biogenesis, the TCA cycle, and then acetyl-CoA synthesis. So first, iron-sulfur biogenesis. A number of years ago, my former student, Dr. Vinay Patel, did a really interesting study in which he showed that iron-sulfur biogenesis is defective in cardiolipin-deficient cells. Iron-sulfur clusters are either 2 irons and 2 sulfurs, or 4 irons and 4 sulfurs. These clusters are essential cofactors in many cellular pathways, including pathways of energy metabolism. And it turns out that iron-sulfur cluster biogenesis is not it's not as simple as you take iron, you take sulfur, you mix them together. It doesn't work that way. It's actually a very complex process that it's very highly conserved, incidentally, from yeast to humans. And it can be seen as two major steps. In the first step, iron is escorted to the scaffold, iron-sulfur scaffold, by this protein, YFH1, or frataxin, which we heard about yesterday. And sulfur from cysteine is gotten from cysteine and escorted to the scaffold by all of these very important and actually essential proteins. And so that's the first step. And then in the second step, the iron-sulfur cluster is transferred to an apoprotein to make the active holoprotein. Again, many important proteins are involved in this step. And there are a number of iron-sulfur enzymes in the cell. So Vinay showed that many iron-sulfur enzymes exhibited decreased activity in the cardiolipin mutant, including the ones here, but I especially draw your attention to aconitase and succinate dehydrogenase. These iron-sulfur requiring enzymes are enzymes of the TCA cycle. So both of these enzymes are decreased in the yeast CRD1 mutant, and Wenja has now shown that there is a decrease, about 50% in Barth lymphoblasts, and also a decrease of even more than 50% in the TAZ knockout cell line. So this defect is clearly conserved. So where do these enzymes fit into metabolism? Here's the TCA cycle, and here's aconitase, here's succinate dehydrogenase. You would imagine if these two important enzymes are defective that there would be deficiencies in the TCA cycle. And among many deficiencies that you could see, or many indications that there's perturbation of the TCA cycle, you might expect to see a defect in glutamate, which actually feeds into the TCA cycle in a sort of anaplerotic step, and also is synthesized from alpha-ketoglutarate. So we decided to take a NMR metabolomics approach to look at metabolites of the TCA cycle in collaboration with Dr. Smithy Gupta at Wayne State University. And here, as we know from many of the really interesting studies that we heard in the last 2 days, there is a wealth of data that you get from this, and how to really focus on the data that will give insight into the mechanism is really key. So even though the metabolomics study is unbiased, still we look for specific predictions that are based on our hypothesis. And we predicted that there would be a glutamate deficiency. Now, when you have all of the data, and you can, which is impossible really to show, but you can see that there are differences between the two things that you're comparing, in this case, the wild type and the CRD1 mutant, and you look for what metabolites cause the difference. So these peaks refer to metabolites that are more prevalent in whatever strain, excuse me, what strain that we're looking at. So here we can see, consistent with the hypothesis, in that there is a much more, there's more glutamate in the wild type cells than in the CRD1 mutant, as predicted. And what is consistent with that, if you grow yeast cells in a stress condition, where they don't grow as well as wild type, but if you add glutamate,